I think I think I'm online again. Let me please check if everything is okay. Yes, I can also hear you. And with that, I'm going to say hi, hello, and welcome again and to another Saturday Microscopy live stream. Today, a special request video. Yes, sushi under the microscope. Why a special request video? Well, yesterday at school, you know that I'm a teacher, a couple of students approached me and said, when are you going to do another live stream again? And I said, well, every Saturday I'll have a live stream. And then the students asked me, well, what are you going to put under the microscope this time? I said, I have no idea. It's always one of the biggest challenges. What should I put under the microscope? You make a suggestion. That's what I told my students. And they said, why don't you put some sushi under the microscope? Here we are. Okay, so I immediately went out to the takeout restaurant and I ordered myself they call this the large sushi set. And I took it along yesterday and here we are. Okay, and looks nice and it was it's 24 hours old. So what I have done, as soon as I was at home, I immediately froze it. I put it into the freezer, now it's been back out again to thaw it, of course, because ultimately I do want to eat it as well, obviously. So it's food, and therefore we have to stay reasonably clean here. And I'll be taking some of these fish and rice and we're going to have a little look under the microscope. Usually at the beginning, for the first couple of minutes, I just want to say hello all around the world so that people have some time to join in. From Vietnam to the United States, Florida, from Germany, Netherlands, Canada, Netherlands, from London, from Northern France, from the UK, from England as always, and from Poland, from the UK, from the US, and it looks delicious. It is quite delicious. Hello from Wales, yes, hello. So the idea is the following today, okay? This is going to be a little bit of a challenge, right? Because I'm going to try to prepare more or less live some of the fish and the rice and the wasabi and so on, and some seaweed, and we're going to put it under the microscope simply to have a look at it. But this does not always mean that we're going to see something really great. So there is always a little bit of a surprise factor involved, as always, but it's a little bit the format of this stream here. If you want to ask some questions, please always indicate this with at Microbehunter or at Oliver. Maybe at Microbehunter is better because then it's highlighted and it's much easier for me to see. Additionally, I got some fortune cookies, and we're going to start off with soy sauce, because soy sauce under the microscope is very nice. However, we have to crystallize it first. So what I've got over here, and this takes a little bit of time, I've got a hot plate. This is my favorite coffee warmer. Which is also suitable as a hot plate. And what I'm going to do now is I'm going to show you how to make some nice soy sauce crystals, and it's going to take a couple of minutes, and for this reason I'm going to start off with that. And then we're going to try, maybe we're able to observe some of the food here under the stereo microscope, and then also under the compound microscope. Okay, so let's do the following. Just for your information, that you're a little bit aware, I've cleaned everything. I've disinfected everything because I've been picking out microbes with those tools here as well. I disinfected and cleaned everything so that everything's nice, food ready, right? So this here is always a difficult thing to open because it's going to be a mess. So let's start off, preparing some of the soy sauce for microscopy. This is actually, I would say, soy sauce is produces one of the nicest crystals. And I hope I'm also able to demonstrate that this time, because there are plenty of salts, and I think also amino acids, maybe also some other things in here. And for this reason, it's slowly starting to become warm. See, there's this, let's put it away. And let's simply apply a small drop of this soy sauce on the microscope slide, and then we're just gonna wait, right? Yeah, so these are nice clean microscope slides. Yeah, this is a fresh tip. Everything's been nicely, yeah, cleaned. So yeah, this is not enough, right? We're gonna apply way more. We're gonna spread this out a little bit as well. Yeah, it's nice because of the all of the dissolved substances. This breaks the surface tension, and therefore it stays nicely spread apart. And of course, now we could wait a couple of hours, and because we don't want to do that, yeah, I'm going to, of course, put it on my hot plate. Yeah, and we're just going to wait a few minutes. And in the meantime, yeah, you can actually see how it starts to crystallize here. Yeah, you just basically just keep watching how it starts to form crystals. And then as soon as it starts doing that, I'm quickly going to put it under the microscope. Okay, so that's one thing. This soy sauce I'm going to put to the side because I don't want to spill anything. And I would say we're just going to start with, you won't believe it, the chopsticks. I'm going to put the chopsticks under the microscope. And indeed, many of the things that I'm doing here right now, yeah, I have not tried before, and it's a little bit, it's starting to crystallize already. Yeah, I don't know if this is, this was very quick actually. Yeah, let's see if, actually these crystals here, they don't look quite as exciting. We're gonna see. Yeah. So, I would like to put a piece of the chopstick wood under the microscope first, because chopsticks, it's also part of the food here. Okay, and I have to also say the following: I'm living in Austria in Europe, and the wasabi is not quite as hot here as in other countries. They are adapting the taste, yeah, of the food, of course, to the local the situation as well. This is now the most dangerous part, and if I happen to cut myself, which is quite possible, I think I'm have to change around the topic a little bit and to put my blood under the microscope. The idea is I would like to see the cross sections here and maybe some of the cells here. So what I have to do is I have to carefully try to shave off. I don't even know if I'm able to do that. I would like to shave off a little bit here, maybe. Yeah, and let's see if this actually works. If not, then I'm going to try to shave it off the other direction, but occasionally we are able to see, this is very difficult. Okay, but sometimes you don't need very much. Let's try it like this here as well. So if you're lucky, you can actually see quite a nice wood structure, but because this is bamboo, it's actually a little bit significantly harder. Okay. It's a little, yeah, let's see. I know, I wanted to start off with sushi, and I'm now actually starting off with the wood. Okay, let's give this here a try first. As always, we have to add a little bit of water as well. And I just also want to make something clear here, because this is a live stream situation and we're always a little bit under time pressure. Sometimes the preparations I'm making are not the nicest ones, right? The point here is that this should serve a little bit as a, how do you say, an inspiration on what you can try at home yourself if you have a microscope. And yeah, therefore, not everything is perfect, obviously. It's more sometimes the things are a proof of concept. I just realized that I did not wash my cover glasses properly. There is still some dirt on here, which is not so good. Okay, and let's give this here a little try, and let's see if yeah. I just see that this one over here, the cut over here, is not quite. This is probably not going to. Let's add a little bit more here. And it's always a little bit of a luck as well if we're able to see something. But let's have a look at some of the shavings of the bamboo here. Yeah, yeah. I get it. Yes, correct. Bamboo, strictly speaking, is not wood. Yeah, it's. I mean, it's still quite hard. It's belongs, of course, to the so-called to the grasses. Look, 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 look. Now the crystals here are nice. That's the next thing to look at. Yeah, yeah. So let's have a look. Yeah, I'm going to now check the. I need to find the microscope here. Oh, here. Oh, where is everything? Here we go. Oh yeah. We're now at 40 times, and at least I can show you a couple of cells. I mean, isn't this what a biology lab is all supposed to be about? A couple of cells. Yeah. And here we go. And we go up. And yeah, that's basically the cross section. That's why I've tried the cross section. And of course, yeah, I'm not gonna try to do too much of a biology lesson here, right? Yeah, but of course, what you see over here, those little circles here, these are of course the cells here, yeah, that you see, yeah. Actually, quite nice. Seems to be reasonably thin. As I focus through everything here, I can see that maybe I indeed managed to shave off one single cell layer. Yeah. So actually, I'm quite happy that at least this little demonstration here, yeah, worked out. Yeah. So I wonder if I've got an yeah, that's the longitudinal one. Remember, I just also did a longitudinal section. I wonder if I'm able to see some of the vascular tissue as well here. Let's see if let's go up here with the magnification here. Yeah. Oh, that's the longitudinal cross section of the bamboo. Yeah. 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 So yeah, there's now a discussion going on. Is basically wood. I mean, the point is the question is now: is it lignified? Does it contain lignin? Because lignin is the chemical substance of wood, and even if bamboo belongs to the grasses, if it contains lignin, which is the hard material, then we can officially call it wood. Yeah, because it's a chemical. Yeah, a chemical thing. Okay, so okay, yeah, this was kind of a little bit of the warm up. Okay, so now we've also seen. Now I can say that I also put some of the chopsticks under the microscope. Okay, yeah. What makes the colors so crazy? Good question. Okay, especially if you've joined in the live stream. I think, but that's important. I've got these beautiful, nice colors. You know what? I'm going to go yet crazier. Look what I can do here. Isn't this nice? Yeah, you can choose, and the reason is because I have a special kind of optics here called DIC, differential interference contrast, which adds color. Pretty complicated optics, polarized light, and prisms and all of this stuff. Generally, I do it with those colors because it kind of looks nice for a live stream, right? However, if you are trying it out yourself at home, then this would be actually the way that you would see it. That is regular bright field now, okay? So these are the natural colors. Yeah, yeah. So it's yeah, that's basically how it's real, right? And with this here, we are adding a little bit of color, and this technique really requires a lot of light. It really eats away a lot of light. Yeah. Okay. Yeah. Thank you very much. Ever smaller, Jesse. 23.8% lignin according to the NIH. So I guess, yeah, so if it contains lignin, then we can officially call it wood. Okay? So we are now going to move on, and I would like to now put in here we go with that's now the soy sauce. Just a second, I'm going to show you this thing first with here. That's the soy sauce, okay? This is how the soy sauce now looks like. Yeah. So because I put it on the hot plate, it evaporated. Let's have a look at those crystals here. Let's have a look at the crystal scope view, and then let's go for the sushi. Yeah. And yeah, I'm going to now use bright field again, so that you see it now how it looks like if you are using it. Yeah, it looks quite nice. Yeah. Looks quite nice. So I removed now also the polarizing filter. So yeah, 4 times objective. And usually what I do, because I like to play around a little bit, usually what I do is I then also dissolve I add again some fresh soy sauce again. And then we can actually see, just check what I'm doing again here on the top. I'm adding some fresh soy sauce here on the side. And you can actually see how it spreads and how it starts dissolving everything again. Yeah. So you can play around with that. Yeah, why am I showing you this stuff? If you're wondering what could I put under the microscope, yeah, this should be some kind of a, how do you call this? Yeah, some ideas, right? Yeah. Sodium glutamate, yes, this how do you call this? It can be maybe it contains also a lot of regular salt, and then also the how do you call this, taste enhancing thing that's very often added also to Chinese food, might be also in here. Monosodium glutamate, which is an amino acid, yeah. Yeah, so or a combination of different things, and because we've got now a saturated, solution, if we wait a little bit longer again and allow it to evaporate, then you can actually see also the crystals forming again, right? Honestly, if you don't know what to put under the microscope, just try soy sauce. It really makes nice crystals, yeah. And yeah, but now let's let's go to the sushi. I would say just for the fun of it, because this is going to be the really challenging part. Because I am a little bit, how do you say, worried, concerned whether I'm actually able to prepare the fish in such a way that I'm able to actually, we're actually able to see some cells and some muscles and so on. So the first thing that I'm going to do now is the following, is I'm going to, because I would like to eat this as well, and I don't want to spill anything over it, the first thing that I'm going to do is I'm going to simply get some of the food here on a separate plate, and the rest I'm going to put back into the fridge. So we're gonna put some, this here, this is gonna be our source of rice and fish. It's the same fish as salmon as we have over here, so I can just leave this one over here. This one is crab. I'm lucky that the whole thing is already thawed up. Okay, we've got some of this this is over here, that's a ginger that I would like to put under here. This is some, I think this red one is tuna, or this one here, I don't know. This is seems to be some kind of a shrimp. You know what I'm gonna do? Let's have a look at the tail of the shrimp here. So I'm going to try it like this. This is not going to be easy to prepare. I don't know. This is not going to be easily preparable. And I don't know, some wasabi, of course, I need. How am I going to get that? I'm going to use, for the wasabi, I'm just going to use my clean toothpicks over here. So you don't see that here. Let's take some of this here. Yeah. I guess, maybe this one here as well. You know, from this here, I'm gonna take a small sample. You know what I'm gonna do? I'm just going to, look what I'm doing, like this. I'm just gonna cut a piece off using my scissors. It's gonna be fine. Yeah, and the rest will go back over here. We're gonna see how this works. Okay, so I'm gonna close it off. And the first thing that I wanted to, let's just put it over here in case I need it later again. First thing that I want to do is we're going to have a look at this, I would say under the stereo microscope, but I just realized that this one here is also quite big. Yeah, I'm just gonna, let's put it under the stereo microscope and let's see how it looks like. First time ever, honestly, also for me, that I'm looking at sushi under the stereo microscope. And the magnification is way too big, as we can already see. Yeah. Yeah. Pooh, too big, not practical. Yeah. Yeah. Not really super interesting, I would say. That's the ginger part here. What's this white stuff here? I don't know. Maybe some rice. Oh. Yeah. Okay. Let's do the. You know what? One thing that I have. I'm going to do now is I'm going to put the easy one first under the microscope, and that is some of the seaweed here. The seaweed is quite easy to observe because it is very thin, and we should be able to see the individual cells. Yeah. So let's do the following. Let's take a small sample of the sea. Ah, look here. Actually, it's. And the rice itself, I'm going to try the following. I'm going to try to stain it or using some iodine. So here it is. That's easy. It's nice and flat, and then we have to add a little bit of water again. So. And again a cover glass. Yes, this seems to be okay. But you never have too high expectations because, of course, we want to only evaluate everything after we've actually looked at it. Wow, this is way too dark. Okay, this is way too dark. Wait, let's look at I don't know, a bright area. Yes, slowly. We should be able to hopefully see the individual cells again, because that's, yeah. So, and now we're at 60 times, and I'm going to open it a little more to allow more light in. And here we go. Yeah. I've honestly had once many years ago, several years ago, I put one of these under the microscope as well. This the red algae here. I could see it a little bit better. But those regular structures that you see are, of course, the cells. Yeah. Okay. Sushi translates to sour rice. Didn't know that, but indeed they're adding vinegar and sugar. Here, that's, I think, a little bit nicer. Yeah. So that's basically how that's the stuff that we're eating. That's the red algae. And as expected, made of cells. Yeah. And not so difficult to observe because the specimen is naturally very thin. Okay. Yeah. So I think the fish itself is going to be somewhat of a greater challenge. Let's go down again a little bit. You see the air bubbles. It's pretty dark, I would say. Bubbles, bubbles, bubbles. Maybe over here. This could be, maybe we're a little bit more lucky here. Maybe I've got a single layer. Yeah. Here, that's nicer. That's what I was looking for. Here we go. Up, I bumped it. That's what I remember. Okay. That's now basically a very thin layer. And here we are able to see the cells much nicer. And here we go. Okay. That's also interesting. You occasionally see that as well. That when you're looking also at plant cells, you see very often that the cells here on the edge, basically are very often, they don't have a color, while the other ones are pigmented. And the reason is that those cells are damaged because they were cut open or ripped open, and then the cell contents disappeared. And the pigment that you see in here, that must be obviously the photosynthetic pigment, because those algae, of course, do photosynthesis. And when you cut it open, then this pigment, the cell's contents will run out. Yeah. Are these cells arranged in pairs? It almost looks like it, at least here I'm at the top, but not only in pairs, but look. And maybe it could be, and again, I don't know so much how they divide, but sometimes maybe it could be that when they divide, right, then there are two of them together, and then they divide again, then you've got four. Yeah, and you see that the individual cells here seem to be smaller, so it might be an indicator that they just have divided, compared to the cells over here, which are larger. Yeah. So, yeah, I think it's also. In case you want to see how this looks in bright field, you see how much brighter it becomes because DIC takes away so much, so much light. That's regular bright field. Yeah, now we can actually also see. We can actually also see a little bit better that the cells here on the edge here, are actually ripped open. Okay. So let's move on. Compared to onion cells, these algae cells seem very small. Is that right? Definitely right. Honestly, onion cells, at least those of the onion skin, are huge. They're really huge. As a matter of fact, you could even see them if you take a magnifying glass. You can see the tiny structures. So they're just at the level of where you can actually see them yourself. And these are much significantly smaller, right? Okay. That's an interesting one about publishing something. It's a little bit of an off-topic question. Please, I would like to answer this. I think that's an important one. Yeah, this is a long name here. Basically a home-based microscopy lab. And if you want to get something published, what do you do, right? And I'm going to be, I'm going to comment a little bit on that. Okay, I'm going to comment a little bit on that. And what I'm going to do now is I'm going to switch back to the desk view. Okay, so that was basically the red algae. The next one is gonna be slightly more difficult. Let's try the ginger here. This is a huge thing here. This has been cut. It does seem to be a little bit more. Here we go. It does seem to be a little bit more transparent. So, I'm gonna try the following. I'm going to just try to put this here as it is directly on a microscope slide. It's going to be way too thick. Okay, this is gonna be way too thick. Let's first put it under the stereo microscope. Here we go. Maybe we're able to see certain structures here. No, not really. I'm going to try the following. Let's turn on the light from the bottom. Actually, this seems to be light from the bottom. Seems to be interesting. You see, it is transparent despite its thickness. Despite its thickness, it is transparent. So maybe let's give it a try. On a regular microscope slide. And let's see if we're able to see this a little bit better. So, desk view. Here we go. This is a huge one, and let's try the following. Look, I've got all these huge cover glasses here. Well, yeah, it has these yellow dots on it, which is also something I was not aware of. And let's see, this covers almost the whole thing here. No, not absolutely necessary. You know what? Sometimes I do the following. I just drop it on directly, and then I'm going to add water. Second step. Sometimes it's easier this way, and then you allow capillary action to do the rest. But it is a very thick specimen. So, yeah, it's probably not the... let's just add more until everything's filled up. or almost everything. So, yeah, of course, this also means that I'm not able to go up very much with the magnifications. So I have no idea what the yellow dots are, seriously. But that's why we have a Saturday live stream. Please, seriously, I do the same thing also in my classroom. Some students have a question, I don't have the answer. I said, I now allow you, even though we have a no mobile phones a regulation in school, I tell them now you're allowed to pull out your mobile phone or your tablet computer and you're able to now research something for me and please share it with the rest of the class. So I'm just going to do the same thing here. Yeah, please, research something for me. So this is now, yeah, let's go down to the four times. Yeah, we need to focus. So that is now the ginger, okay? Look at this. Look, that's now using the 4 times magnifying objective, okay? Using bright field, so no color. That's interesting. What are these dots? Maybe some I don't know, some oil droplets from I don't know. No, but there is. Look at this. Isn't this interesting? Now we can actually see all of the cells here, the cell walls. Let me quickly go over here at the edge where it's much thinner. So here it's much thinner. You still have those. That's interesting. Yeah, I'm now with my 10x objective. Here it's very thin. So 20 times. This is pigmented. I wonder if these round things are really part of the ginger or not. Could it be starch? There's only one way to find out, right? There's only... Is it starch? Honestly, I have no clue. There is only one way to find out. And what way is that? Ta-da! You cannot... Can you read this? Iodine. Yeah, let's go for it. Let's try it. I have really no idea. So what I have to do now is I'm going to over here in the speaker. I'm going to, where is the desk view over here? Yeah, let's look. When we do iodine staining, we gotta be a bit careful. This iodine is very strong. It corrodes. I do not want to get the iodine in contact with any of my microscope parts. So what I'm doing right now is I'm going to take some of this ginger over here, right, that I still have, yeah, over here. I'm gonna be adding some iodine. I'm gonna stain it and I'm going to rinse it again. Okay, that's what I would like to do. Is, let me see. Yeah, that's... No, is this even the ginger? This is not ginger. This is part of the fish. Or not. You know, that's the ginger. Oh, gee. Here it is. Okay, so... I need a very thin part here. So let's just do it like this. So let's, it's in here. Some iodine goes in it, on it. And yeah, let's allow diffusion to do its action. And then I would like, it does stain already. Some parts do stain black now. And then we're going to find out which parts will stain black, because iodine and starch do a, the classic iodine reaction, and therefore we should be able to see which parts actually do contain starch here. Yeah. So what I need to do now is the following. I'm going to now take it out again. And I would like to take it out again on a slide. How am I going to do that? I'm going to actually, where is it? I need another little beaker to actually, I'm going to take this here. Yeah, you clean this off. So, and I'm going to rinse it now in a little bit of water to remove any excess iodine. I don't like, I don't like it. That's why I'm also not using the tweezers, because iodine is corrosive and I do not want my metal to corrode. You see, it does stay black. So we've now removed any excess iodine. Very nice. And we're now going to put that again under the microscope. And now basically I don't mind picking it up so much because the iodine is very dilute. Here we go. Cover glass goes on top. Cover glass goes on top. Yeah, it's already actually surrounded by water, so that's not so much of an issue. Let's add more. This is a very thick specimen. But we're now going to find out which parts over here actually did stain black. So scope view, yeah, that's the unstained one. Yeah, from before. And this is now the iodine stained one. Ooh, way too dark. Way off. Ah, very nice, very nice. And we have already an answer. Do you see those tiny black dots in here? Those tiny black dots, these are starch grains, and those large orange ones evidently were not. Because it does look a little bit like those large bubble-shaped structures that you see here. Yeah, these seem to have been these orange dots here, and those dark dots here, that's starch. So that's the starch storage, yeah, of the ginger root. Yeah, so it actually worked quite nicely here as well. I mean, if you do this, I've done this here, a lot here. Oh my gosh, look at this. There seem to be different parts of the, a lot. Different parts, I'm gonna really go up. Different parts of the root of the ginger actually do seem to store starch differently. If you do this with a banana, for example, beautiful starch grains, potatoes, I don't have to mention because that's the classic. Yeah. Is it the same iodine that you put on wounds, or the point is the following: the ones that you put on wounds is much more diluted. This one is very concentrated stuff, probably not suitable for wound treatment. Yeah, this is way too concentrated. Yeah. But yeah. But you see here, that's yeah. But would it work if you were to put it on wounds? Yeah, but I probably wouldn't, because there's the danger maybe that you're overloading your body a little bit with too much iodine, you know. Yeah. Okay, that we have answered now as well. Okay, indeed, that was the ginger that we that goes along with the sushi and the sashimi, and yeah, and there are some starch grains in here. Yeah. So very nice. Occasionally, I have not been reading now many of the comments, so I'll quickly do that. But there were not so many comments directed towards me. There was one comment about publications. Yeah. Could be the pickling. Yeah, exactly. This orange. Yeah. What camera am I using? I'm using a Canon D90 camera. It's a DSLR camera connected directly to my microscope without intermediate optics. I have got HDMI out, and this HDMI out from the camera, full HD HDMI out, I'm converting to USB using a small adapter. Yeah. However, the image quality and so on is not necessarily just one of the camera, right? It depends also on the optical system that you use and other things. The only thing that I really like about my system, because you're already asking me about the cameras, is that the field of view that I'm getting is actually wider than looking through the eyepieces. Yeah, so it's filling. Actually, I see more, of the specimen through the camera than actually through the eyepieces. Yeah, and I've got pretty decent eyepieces. Yeah, so yeah, let's move on. So that was the ginger. I do have to check every now and then a little bit on the and now let's try to move on a little bit. Yeah, let's try to move on a little bit. We can always come back to some others later on. Let's now move on and look. Should I try this? The wasabi. How am I going to do that? The advantage of the wasabi is that it's very soft, so I'm able to press it into a thin layer, into a thin film. That's called a squeezing specimen or compression specimen. It's like, I need to make sure that the iodine is closed. The thing is the following, that in order for you to observe something under the microscope, the specimen has to be sufficiently thin and transparent. And this is not always the case, but with wasabi as such, it's fairly easy, because we have to make sure that we're really making it very thin. So what I'm going to do now is I'm going to take a tiny amount here. And I do not have high expectations here, because it's a very homogeneous thing. But maybe, hopefully, we are able to identify some plant material. So I'm going to now press this. Now we have the problem that if I press down on here, I'm going to break the slide, because the surface is soft. Yeah, it's for writing, it's very soft. So what I'm going to do is I'm going to put it here on the table. I'm going to put some tissue paper over it. Now you see it's a little bit upside down, right? And now I'm pressing down vertically without any horizontal shifting. Yeah, and I don't know. This is how it looks like. I don't know if it's thin enough, but we're going to give it a try, and then let's see if we're able to find some plant material. Square scope. Yeah. It's too bright. It is still too dark. This isn't... I don't know. There's some bubbles as well. I mean, it does look interesting, for sure. But those patterns here are because, maybe I have to mix it with a little bit of water. Yeah. I don't know this, but there seems to be something there in any case. What are these weird dots? So that is wasabi, which is indeed some horseradish that's plant. Look, can you believe it? Found some larger pieces of this horseradish. I see cells. Now I'm happy. Let's add some color. Yep. It's ground, because it's kind of, this wasabi is this ground up root, and apparently I found now a little structure here, where I'm able to see some of this plant material. Yeah. Very nice. Evidently there are larger structures still present. Do you have a video on how to clean your microscope lens? Yes, as a matter of fact, a couple of years ago I did make a video on how to clean microscope lenses. You have to use lens paper, and you got to be careful to only clean. Make sure. Look, most likely, if it's dirty, your microscope optics, then it's most likely the eyepiece and not the objective, because unless you got in contact with the specimen, it's usually the eyepieces, because the eyelashes they touch the eyepieces, and there's always some grease and grime. Yeah? Okay, that's the wasabi part, and I like it. I like it because I did not expect to actually find still plant structures in the wasabi that are that large. Yeah? So that's really cool. But you know what I'm gonna do, because I like it so much? I'm gonna do another one, and I'm going to now mix it with a little bit of water, and I'm going to try to, yeah, spread it apart a little more. And then later on, I still have to fish. Oh, you don't see that. I put, where is it? Ah, yeah, here. I put a drop of water here. A little bit of this tiny amount of this wasabi now. Let's see if I'm able to completely dilute it. Oh, wow, it's still very dense. Still very dense. Let's add a little more water here. And a cover glass goes on top. Let's see. Blip. Oh, yeah. It's maybe a little bit better. I don't know. Honestly, let's have a look at this here, whether we're able to see a little bit more. Hope view. Yeah, I'm using tap water. No, not chlorinated. We've got pretty clean tap water here. It's drinkable water. Yeah. And this is now the wasabi. Wow, what a mess. But we are able to see the individual plant parts here. Yeah, here, let's find some. Yeah, so all of these larger fragments over here are evidently parts of the plant, but we have to actually zoom in a little bit more to actually see maybe the, yeah. Let's go on here. Yeah. What a mess. Gee. So many other particles in here. Yeah, maybe this one could be. Let's see if this is better visible. Yeah, not so much. Huh? But what we are able to see here in the background, all of those little particles that are floating around, they're vibrating. Of course, that's so-called Brownian motion. They're not moving on themselves, but essentially because of the thermal, everything is temperature, therefore they're kind of bouncing around those tiny little particles here. Yeah. Is 70% alcohol okay to use? I ran out of lens. Solution number one. Because you're asking, but maybe no, you want to use a higher percentage alcohol because 70% alcohol contains 30% water. Number one. Number two, you should not use commercial lens solution because it is not pure alcohol and sometimes there is a residue left over. So what you should use is you should use actually an alcohol. You always should do that what the manufacturer recommends. But what I've heard is that so-called alcohol to ether. Yeah. So this actually now looks very much, those things here that you see, those things here, they look very much like starch grains. Okay, so what I'm going to do now is, because of this, I'm going to do the following. I'm going to now try to actually verify if this is indeed starch by now mixing the wasabi with a little bit of iodine. Okay, and then let's have a look at some of the fish as well. Where's just a second? I need to get the desk here again. So I'm just going to recycle this slide. Okay. I'm going to add a tiny amount of iodine. Yeah. Yes, ethyl. Yeah. Just a second. Nah, this is too much. I have to really do the following now. I'm going to take now a small amount here. Here it is. Okay, let's do it like this. And I'm going to mix in a little bit of the wasabi now. Ah, look at this. It immediately turned black. Yeah, plenty of starch. Plenty of starch. Yeah, you can already see the starch grains. So, yeah, almost like this here. So very quick observation under the. Now I have to be a little careful because there is the iodine still in here. Yeah. Yeah. Let me see. Here we go. Yeah. So that's now the wasabi with iodine. You see all of those dark dots here. That's all of this starch. Yeah. Yeah. So we now also know plenty of starch in wasabi. Okay. Yeah. Okay. Uh, yeah, that's the whole. Yeah, exactly. All of the roots that are used for human consumption will contain a lot of starch. Of course, that's the reason. Indeed, because that's the reason why those plant make those roots as a starch storage. Same with potatoes, for example, yeah, and carrots and so on. Of course, yeah, why do they have all of these? Well, because of course that's how they are storing the energy in the form of starch. Yeah. Yeah, it's all starch all the way down. Yeah, basically, this reminds me of someone who said that. It's all the way down. Wasn't this Stephen Hawking in his book Brief History of Time? He says, "Well, it's turtles all the way down." Okay, so let's move on. So we looked at this. I want to see. Wasabi. This is a little bit. This how do you call this? This exoskeleton of a shrimp. How is that? Let's have a look at this. It's the, yeah, it's very thick here. So what I'm gonna do is I'm going to cut off even more. So we're gonna look at the it's the chitin here, and let's go. So that's basically from this, yeah, from a piece of shrimp. I'm going to take new fresh. Okay. So here we go. Well, what was this? Yeah, it's turtles. How did that go? It's basically about the structure of the universe. And then Stephen Hawking said the story in his famous book, A Brief History of Time, where this old lady in one of the lecture halls says, well, actually the Earth is resting on turtles, and it's turtles all the way down, right? Yeah. Wouldn't the stereo microscope not be a better choice? Probably. You know what? I think yes. Okay, so it's still covered with water, so we're gonna put it also under the stereo microscope. Yeah, but let's just because I already have it here. Where is the? It always takes me some time switching around the. Ah, okay. Wow! Wow. Okay, so that is now the shrimp. Okay, the one of the that's interesting. What are these dots? Huh. Yeah, cool. That's it. That's interesting. So let's move to a higher magnification here. I'm going to remove the. Okay. Sometimes it's interesting to look at the stuff that we're eating. Yeah. Ah, yeah. If it's specifically made for microscope objectives, then I would trust it. Okay. But generally, what they recommend, basically, we're talking about lens cleaning. Generally, what they recommend is that you do not use those cleaning solutions that are made for eyeglasses, for example, or obviously, or windows and so on. Huh? You have to use a fairly volatile solution, basically one that evaporates very quickly, because otherwise there's the danger of the liquid actually flowing into the cracks of the objective. Yeah, okay, so that's the shrimp. I don't know if I'm able to go yet higher. It's a fairly thick specimen as well. I don't know. Maybe these are kind of some pigment cells, or I don't know. Pigment deposits. Yeah, fleas on the back of fleas. Yes. Okay. So this was that's the shrimp. Interesting. By the way, some of you are talking about coffee, right? um, Coffee is also quite nice under the microscope. Yeah, you see actually the different coffee particles. The question is the following: what are those regular patterns on the shrimp? I have no idea. Maybe some kind of pigmented cells for the pigment. I have no idea. Seriously, for me, it's the first time that I actually also see, a shrimp, an exoskeleton at least. Yeah. So, I mean, which doesn't surprise me. If it wouldn't surprise me if maybe this is somehow responsible for the reddish color. Yeah, but let's do the following. Let's have a look at the shrimp using the stereo microscope. So I'm going to just take the other piece now on a slide, and I'm going to switch over microscopes, stereo. I'm going to use now top illumination on a slightly darker background. I'm going to zoom out as much as I can. Oh, wow. There's some hair over here as well. Maybe we can also have a look at this. Okay. Look, I mean that might be interesting to look at as well. Okay. Here you can also see those dots. Oh yeah. So I don't know if this has any specific function or not, or if it's just some kind of to give it some kind of pigmentation. But you know what? Just having a look at this, I'd like to see some of this, those little tiny hair a little bit more under the compound microscope. Chromatophores for making pigment. Yeah. Okay. Thank you. Your questions are here again. Just maybe I'm not gonna stress myself. I want to do the microscopy first, so we're gonna see how far we can actually go with the questions. But very quickly, I'm going to drop a cover glass again directly on here. And we're going to see how this looks like. I need to always get a... You see, you can... I just, before the start of the live stream, I washed them. So what I've done is, first of all, I rinse them, then I add a little bit of a strong vinegar to remove any water stains, and then I rinse it again. It's reasonably clean. Nope, no, that was not the one. No iodine. I want to have regular water. This is way too thick. Yeah. But it's okay as long as the part that you want to look at is submerged in water. Yeah. So, and this is just for the tiny little hair, which do not even seem to be so tiny. So, oh yeah, I need to go to the scope, microscope. Here they are, those hair. Oh, there seem to be hair on the hair. Oh, look. Huh. The air bubbles come out. Isn't this interesting? Let me... Yep. Interesting. Yeah, I wonder what the function of those are. It kind of looks cool. Bloop, and the air bubble came out. Huh. Let's go down a little bit again with the magnification. Let's add a little bit of color. Yeah, color and contrast maybe. Oh yeah, look. Somehow all of the air is being forced out by the capillary action of the water. But it kind of shows that those tubes evidently are hollow. It's a little bit, that's one of the fun things about microscopy. It's kind of like you're going a little bit on exploration. You just put everyday stuff under the microscope. I remember, I mean, if you're interested in this was several years ago, must have been, I don't know, 5 years ago maybe, or even longer. You gotta check my other channel called just this. Now you're on the sister channel, but in the other channel I put some a Big Mac ones under the microscope. I did when I put the Big Mac under the microscope. Some people did not like that so much because they said that I'm not being very serious. I should not go into that direction. Should be stay a little bit more in the serious type of science, which I get. But then on the other hand, I do kind of like putting everyday objects under the microscope as well, and it's the food that we're eating, simply because it kind of connects differently to your immediate environment. Yeah. Yeah. Huh. Yeah, silly science is fun too. Why are the air bubbles in the end? The air bubbles, because we've just seen, look, I can just zoom in, maybe I can see a better one. Because, over here, let's have a look. Because those little hair seem to be hollow, right? And look at this one over here. Do you see there is air in here, and the air goes out and makes those air bubbles here at the end. Yeah. Yeah. Yeah, so it's quite unexpected in that sense. Yeah. So that's why there all of these here. So let's move on. Yeah, I still have a rice, crab, and salmon fish. Rice, crab, and salmon. And we're just gonna try that as well, I would say. Let's go with the easy one, the rice one. Okay, rice is fairly, and I already put rice under the microscope once. You see, cooked rice, and generally also cooked potatoes. I mean, they still have the starch grains, but they're pretty large and they kind of swell up and they're large and soft. And we should also be able to see that. And I'm just going to put all the slides that we have over here. So what I'm going to do now again is the following. I need I'm going to use this iodine thing here. I'm going to take a rice grain. Ooh, this rice is already pretty hard. Just realized. And let's put it into the iodine. Let's see if you're able to see this. And boop. Yeah, now it touched this here. And we're now going to do the following. We're going to take it out. We're going to take it out, the rice. We're going to wash it a little bit because I want to get all of the excess iodine away. And this is going to be difficult. Just realized. So I can now take this here, put it on here. Here it is. Cover glass goes on top, and now I don't know if I'm able to actually compress it because this rice seems to be also already fairly hard. It does work. So we're going to take again the edge of the table here. Maybe you're able to see that. What a mess. Oh, look. I dropped it on my table. Yeah, maybe it works. I don't know if... So, this is how it looks like. Okay, so that is the compressed rice grain stained with iodine, and it goes again under the microscope. Scope view. And let's make it brighter to refocus. These are huge grains. Okay, let's go down with the magnification. Yeah, this is huge stuff. Huh. But it does appear that those starch grains somehow seem to be clustered. Yeah, not a lot to see. Let's have a look. I don't know. This is, yeah, those starch grains, yeah, seem to be quite clustered. And if you actually take, for example, raw unboiled rice, and if you wash this rice and you collect, and if you put this rice water under the microscope, then you're of course going to see the starch grains much nicer. Here you see just those regions that are black. That's basically starch. Yeah, not super exciting. Huh? Yeah, so I'm just reading the comments here. Ah, yeah. So yeah, so that's rice. Not super exciting. What I have, however, seen is the following. I've got a commercial slide of a wheat grain similar to rice, which is beautifully sliced in thin sections, and you can actually see the different parts of the seed of the grain, and then also the starch grains quite nicely. Now this, because it's boiled rice, obviously, yeah, the starch has kind of swelled up, right, and you lose the original structure. So let's move on. Let's move on. So let's look a little bit at um, at the crab. I don't know. There is also this artificial crab meat. I don't know if this is natural or not. Yeah, but the question is now how we're going to deal with that. This is going to be a challenge. I have to somehow get this sliced in such a way that I'm able to put it under my... Maybe this red part here would be kind of interesting as well. I don't know, maybe you can try it like this. I don't know. Here there's this part here. I don't know if... yeah. Again, this is all... What I'm doing here right now is all trial and error. So let's put it again over here on. Yeah, same story. Some liquid. Oh yeah, let capillary action do the rest. This is still very thick, I think. So here we go, and I'm going to do the following again. I'm going to press down a little bit, just to make sure that it's a little bit more flat. And I lost my tissue paper. Ah, here it is. Okay, I'm just pressing down again on the table here. Yes, I think it's a little bit better. So I didn't just show you. So here we go. Now it's much, yeah. And it goes again under the microscope. So this is now some crab meat. And that's and I put the red outside part of the crab meat. This is how it looks like, and it's honestly super boring. Because normally if you put muscle meat under the microscope, and I don't know if it's real or not, sometimes you can actually see the cellular structure quite well, but this is just, again, a homogeneous mess. And rather disappointing. Are there any regular structures visible? Actually not. Or those droplets that you see here, most likely these are all fat or oil droplets. If it's muscle, and I was actually hoping to see some muscle fibers. but this actually looks a little too. But I don't know. Maybe I am able to pull some of the, because it's kind of scratched off some from the surface, right? So maybe I am able to do the following. Maybe if I cut it differently. Maybe if I cut it differently, maybe it's gonna work better. I don't know. Let's see. I'm able to. I mean, it does look fibrous. Okay. So let's and I don't know, is it real meat? I don't know. Let's find out. Let's put this one here again. So like this. So now I did not scratch something off from the surface. So I'll compress it now between my thumb and finger very carefully. Yeah, it was very soft. And let's go with this one over here. So, and scope view. This looks very, very homogeneous. Very homogeneous. It was actually, you see when you put meat normally under the microscope, you're supposed to see those muscle fibers and the so-called light and the dark bands. So I'm not able to see anything like that here. Yeah, so that's how this crab meat looks like. Let's just try. Let's go up much more with the magnification. Maybe there's something in there that I'm not able to see otherwise. But it looks very uniform. And actually not a lot of structure here. Okay, let's move on. Let's have a look at the salmon, at the fish. It's actually the this, I hope, should be the highlight. Now it's embarrassing if there's nothing to see here. So, and what time is it? Yeah, 1 minute. Yeah, it's fine. So let's have a look at some of the things here. So I'm going to now try to get some of the salmon out here. I'm going to dissect now the whole thing here. I mean, is this salmon even? I'm almost already blind. I need to put on my glasses here. Yeah, of course it is. Yeah, why should it not be? This is now the fish. Let's try to take a little sample of the fish here. This is very soft. This should be also more easily. So here we go. And now again. And let's have a look how this looks like. Again, no water right now. This was very soft. No, okay, and let's see. Maybe it's gonna look similar like the crab. I don't know. Scope. Okay. Oh, yeah, we do seem, okay, forget about the movement here. We do seem to see a little bit more of this fibrous structure of muscle fibers. This is now basically, yeah, that's not bright field. Let's have a look now. Maybe I'm able to find an area where there is, maybe this one, I don't know. It's very, it's way too, yeah. And of course, that is I mean, this is where the biology teacher now starts to get excited again. This is where the biology, because this is exactly that what I wanted to show you, and where it's difficult to see, but actually not so difficult to see. And yeah, this is 60 times. That's very fine. And I'm going to now show you the thing here, because that's the biology part. And this is actually what it was kind of shows that this is real fish. You see those fibers here. Do you see that there are these slight, very fine stripes, right? Yeah, very, very typical. It's called striated muscles or skeletal muscle. And this is called the light and dark bands. Difficult to see here a little bit because they're so fine and small. But actually, yeah, here again you can see them. Yeah, it's kind of like it's kind of vertically striped, right? And that is an indicator that we are looking at real meat here. Yeah. So that is and that's something that I was at least not able to see in the crab meat. Here I am able to see that. So this basically I can tell you here, this is real fish. Oh, no kidding. Yeah. But let's do the following. Let's remove the thing here. Let's go close the condenser all the way. We're still able to see it. Yeah, you see those tiny little stripes going back and forth. Yeah, these are the striations of skeletal muscle, right? And that's essentially something that I'm getting excited about. Here are lots of them, right? Yeah. Why am I getting excited about that? Because that's also stuff that I have to teach in school. Yeah, the structure of skeletal muscles is called the light and the dark bands. And you find that you find it in chicken, you find it in fish, you find it in mammals. Yeah, and this has been already known for a very long time, right? And I think it even goes back to Anthony van Leeuwenhoek, one of the first microscopists. Yeah, and at that time they thought it looks like a spiral. Yeah. And the thing that they discovered, those early microscopists, they were really surprised by one thing, and I think that's one of the big early realizations of biology is that if you look at the meat of a whale, the biggest mammal under the microscope, you find those things. And if you look at a fly, the muscle of a fly, a tiny insect under the microscope, you also find those things. So this kind of showed, it was one of the early discoveries which kind of showed that, wow, all living things have certain similar structures. And not only that they're kind of similar, but the second thing is that they're surprised, even for a big mammal like the whale, the structure ultimately has the same size as those of a fly. So basically the size of the overall organism does not have an influence on the microscopic size. Oh, thank you very much for the donation. Thank you very much. Yeah. So this is simply something I wanted to show you, and this was a little bit my hope that I was able to kind of find those striations here as well. Honestly, I don't know if you've been following along my videos. Every now and then I always like to come back to those structures here. Also, for example, this is a slide of a flea of an insect where you could actually see those. Yeah. And smooth muscle, correct. Smooth muscle, that's the muscle that you find, for example, in the digestive system, which pushes along the food. It is not striated. Yeah? So that's why these are voluntary muscles and so on. Yeah, they are striated. And that's basically what meat is, right? We're eating striated muscle. Yeah? And here very nicely. Here we see it quite nicely. Yeah. So. So this is quite and I personally, it's my view. I always like a little bit this how do you say slightly the biological history of those discoveries. Every now and then, nowadays with the internet, you can find many of these old books, and you can find the scans. It does take you a little bit of time maybe to find the right places, but then you can actually read the original descriptions, right, of the people who then written down. They were so surprised that essentially the structures of those striations in this case, not only can be found in all mammals and flies, but they actually also have the same size, even though the animal itself has such a big size difference. Yeah, so this is a real thing. Yeah. if the crab was real, this is what it would look like. Also, yes, thank you. This was actually the point. Yeah. So this is why I had kind of my doubts. I'm not 100% sure, but kind of, I know that there is this artificial crab meat as well. But this was kind of the test of whether, I would be able to see this, right? Yeah. So what I'm going to do now is the following. There were a few questions. Let me see. I need 10 minutes. Don't worry, I'm not going to throw away the food. I cleaned up everything properly because that's one of the things as well that I want to minimize, of course, also food waste. But here are a couple of questions. Maybe there was one question also about publishing. Would you want to? Yeah. If we have a home-based microscopy lab and need to publish our microscopy finding in an academic journal, what would be the reputation? Can we publish our findings if a home-based lab? I will tell you the following from my hearsay. There have been apparently, I mean, I'm a school teacher, there have occasionally been some, school projects where students with their teachers did some research, some basic research, and they actually got it published. This is something rather unusual, not unlikely, but it depends all on the so-called on the impact factor of the magazine. So there are certain science journals that essentially, you have to either pay a lot of money to get it published there, or they have very, very high strict requirements. And what happens nowadays very often is that essentially people and all the universities start to go in the direction of open access publications. So very often they either have their own magazines or they publish the stuff on their own websites. Even that is possible. So generally in most or in many cases, the discoveries that you make at home in a home-based lab are not of that, in most cases, importance that you can actually get it published in a very important magazine. So for example, if you discover a new organism somewhere, you probably have to do a DNA test to verify that, right? So the standards are quite high concerning this. There was once, and you can download all of those, unfortunately in German, for those of you who don't speak German, but there is a German magazine called Microcosmos. There was a German microscopy magazine called Microcosmos. It was a classic. All of them can be downloaded for free as a PDF. Unfortunately, for several years, they've not published, they've not made new editions. But this was one of those nice magazines where people were actually sharing, advanced hobbyists were also sharing their discoveries or their documentations with the community. So that's one of the things. A couple of years ago, I think over 10 years ago, I myself decided why don't I make a free downloadable PDF magazine. I did that for a couple of years, but this was a lot of work. Yeah, and this was before AI. I didn't have any editors and so on. So nowadays you could probably get the text checked and edited over AI. If you are interested in that, you can download that as well. It's on my website, microbehunter.com. You can go to you can download all of those magazines, right? So this is, but generally, that's the thing. If you really, it depends really very much on the importance of the discovery, but unfortunately, very much those the well-known magazines, they're quite expensive and sometimes difficult to get stuff published in there, right? So so that's the wood discussion was here. I wonder which fish can be eaten as sashimi and which cannot. It depends on the bacteria, I assume. That's a very interesting question. The point is the following, it's raw fish, right? And raw fish generally is an issue, but what I heard is that I've rarely heard with the salmonella infections and so on with sushi and sashimi. It's interesting. And I claim the following, because if there were a problem concerning bacteria, it would be a real problem, right? So I think they're really checking this stuff very well, because otherwise we've got a real problem, right? Because fish actually can be quite dangerous. Yeah. So the algae cells seem to be quite large. If we have home-based microscopy lab and need to publish, I just talked about that. Yeah. What would be the reputation? I mean, it's it's called the impact factor. And honestly, nowadays, this sounds a little bit harsh if I say that. With a microscope alone, you're barely able to make it's not likely that you're going to make able to document it in such a way that you can get it in most cases published, I claim. I mean, for example, if you want to discover a new organism, you have to do advanced staining, maybe possibly DNA analysis and so on. And the question is it really worth it, right? I like to be convinced of something else as well, but from what I've experienced in the biosciences, unless it's some kind of a hobby-based magazine, right? Which also accepts it's unlikely to get it published in like in Nature or Science and so on. It's the iodine itself is I think pretty much the same, but it was a different, way different concentration. Okay, that's again the starch question that I already talked about. Okay, so what I'm going to do is I'm going to quickly go through the how do you say your comments again. Okay, if there's anything that's directed to me. Okay, C# and C++. Yeah, I read some of the volumes of your magazine. I can see why you stopped. Yes, this was a lot of work. This was before I started to make YouTube videos. I'll tell you a little bit about the history. If you want to know how the magazines look like, please download a few volumes. At that time, long time, when I was much younger, I was generally always interested in the sciences, of course, biology, but at that time, of course, also astronomy. And I was always a little bit frustrated that there are so many astronomy magazines out there and nothing about microscopy. So I said, we need some kind of a print magazine, right? And I wanted to have some paper printed, PDF, whatever. So many years ago, I kind of started this and people sent me their articles and pictures and I kind of briefly proofread them and alone. I didn't have any help. I made a layout and then I basically made them available for free download over PDF. Was a lot of work. However, also the quality was a little bit a problem because I sometimes made mistakes myself, some editing mistakes and so on. So I could also not guarantee a sufficiently high quality. There was no team behind me really, right? So this is a little bit was an issue. So this is nowadays, I don't know, maybe I could pass the text through an AI grammar or spell checker or so on. Spell checking was not so much the issue, but grammar and style. So this was a lot of work, right? For this reason, I decided it's easier to make videos, and that's why I decided to open a YouTube channel back in 2018. And that's basically where we are right now. So I'm just going through the comments again here. Okay, I guess I pretty much talked about all of the comments. As well. By the way, I have to say the following. That's also one of the things where I would like to request the help of the community. I'll admit to something. Over the last couple of years, I've been collecting email addresses for a newsletter. And up to now, I've got quite a decent number of email addresses together. You can subscribe to the newsletter over the website. However, I have not sent out a lot of newsletters yet. Very few. Also, one of the reasons is because I have to also create content for the newsletters. But what I want to do now is I want to send out also maybe once a month, maybe twice a month, I don't know. I would like to send out also a quick summary of the videos and the live streams that I made over the past month, including links. Okay, because I did get the information from co-workers, colleagues in school and other people that they basically do not get the videos recommended anymore. So even if they subscribe to the channel, it's not recommended on the homepage anymore. So YouTube algorithm is changing. So if you want to kind of be informed, on a regular basis of the previous videos over the last month, then if you subscribe to the newsletter, then I'll be sending out these things, right? It's just basically something for convenience maybe for you. Okay, so I'm quickly going to say AI is an issue. I know. Okay, that's one of the things that yeah. I've just recently read an article in one of the science magazines that they were testing how is it possible to use AI to make microscopic images, and which looks like a microscopic image, and even that is possible nowadays. Yeah, so this is a little bit, I think the new reality that we are in right now. That with AI, it's possible also to, to copy, also images from under the microscope. Yeah, so I wonder where this whole thing is gonna actually, lead us. For those of you who basically just joined in a little bit later, and who have missed the beautiful sushi set from the beginning, which I think I'm going to start eating now for dinner. I've basically put shrimp, some crab, some rice, all of these things here under the microscope. And now we know a little bit better of what we've been eating, what I've been eating over the last, I don't know. Yeah. And the lemon. Okay. How do you put the lemon under the microscope? You know what? Actually, I have an idea. As a last thing, it's gotta be the lemon. Is this actually, honestly, a lemon on sushi? Is this actually something that you normally do? I don't know. Lemon actually is a typically, I don't know, you put it on salmon, of course, but normally. And what I'm going to do is, I'm not going to squeeze it out, and I dropped it. I'm actually going to try the following. Let's try if this actually works. Can I do this? This is so tough. This is super dangerous. If I cut myself. This is actually do you actually know that this is a fresh blade? I thought it was a fresh blade. Well, of course it's a fresh blade. Why is it so difficult to cut? Basically take home message. If you want to cut something, make sure that you're always cutting it like this, so that the edges here of the slice are very thin. Okay, so let's do that here. And and then last but not least, before I actually do say goodbye, there's another thing I want to put under the microscope. I just realized. So let's put this here. We have to add water. Let's if we do that, we got to do it properly. And without water, the refractive index issue we have. And where's this here? So I have to add some water. Without water, we can't have a proper refractive index. I now lost my cover glasses. No, here they are. So let's see. Do lemons have raphits? These are crystals. I don't know. I'm right now just putting the how do you say, the skin of the lemon here under the microscope. Let's see. If I actually was able to cut it very thinly. Wrong microscope. So here we go. It's too bright. Okay, that's the edge. Yes, at least. Let's add some color because it's so nice. Yeah, that's what I meant. You have to kind of cut inwards and outwards, and you try to, And here, right on the edge, we can see ourselves. See, now I touched the. Too thick. Here we go. So I have to use the 20 times objective, and of course here we can see here on the edge where it's very thin, we're able to see the individual cells. Possibly fat droplets or oils, because in the skin of of a lemon there are these oils. Okay, so that was the lemon, and you know what I'm gonna do now? I'm going to do the last crazy thing here before I forget. Where is the desk view again? Look. The restaurant yesterday gave me this here. Of course, a fortune cookie. Starch. Of course. That's under the stereo microscope. Let's open it up, and I have to adjust the other. Yeah, that's the fortune cookie here. Of course, this is not, no cells over here, obviously, because it's made of flour and I don't know what, right? But let's see what's in here. No, I need my glasses. Oh, it's even in English. Oh, in several. Okay, what does it say here? Okay. Oh, yeah, of course, this is international for also. Love is the spice of life, sometimes sweet, sometimes salty. Yeah, and then appears to be in different languages. In German as well. And I don't know, yeah. But actually the translation, is not exactly the same. It doesn't matter. Okay. Yeah, so I think I'm just going to call it a day for today. Yeah, hope that it slightly unique, different and slightly different things that I've done today. If you have any suggestions of what you want me to put under the microscope, please please comment, and maybe write it into the comment section here. Italian, so it's not exactly the same translation. Same in German. But I think I'm going to just call it a day for today. I wish you all the best, and I think I'm just gonna see you again next week. And I think I can confirm that this fortune cookie is also made of starch, because as you can see, it turns black. And this means, of course, what did we expect? Of course, it's starch. Okay, I wish you all the best, and hope that these little weird experiments today and observations today kind of also inspire you to do a little bit of microscopy and put things under the microscope. And thank you again for the donation, of course. Thank you very much. Will be used for microscopy. And I wish you all the best, and hope to see you again next week. Okay, bye-bye.